The ligation of Okazaki fragments. The ligation of Okazaki fragments is the final stage of lagging strand synthesis, a key part of DNA replication.
What are Okazaki fragments and why do they occur? Okazaki fragments are short DNA fragments attached to RNA primers. They are named after scientist Tsuneko Okazaki, who first discovered the fragments when studying DNA replication.
Polymerases can only work in one direction when copying a DNA strand, so can only copy one strand. Therefore, the lagging strand is copied in a series of small pieces, including the Okazaki fragments, that need to be linked up in a continuous strand. Okazaki fragments attach to the lagging strand of DNA during a process of DNA replication. In order for the DNA to fully replicate and carry out its functions, it is necessary for the fragments to be synthesized together.
Broken fragments in DNA can cause problems in DNA transcription and replication, and even sometimes lead to serious dangers. To prevent these dangers, cells use ligase to prepare broken DNA and glue together strands. Ligation can be described as the process of joining two nucleic acid fragments catalyzed by an enzyme.
Ligase is an enzyme that joins breaks in the phosphodiester backbone of DNA that occur during replication and recombination. The ligase uses a cofactor molecule, shown in red, for power and a special lysine amino acid, shown in purple, to perform the synthesis. Human ligases use ATP as the cofactor, but other organisms use different cofactor molecules. For example, many bacteria use NAD to fuel DNA ligation.
In Okazaki ligation, the lysine in the DNA ligase forms a bond to the phosphate in the ATP, holding onto the AMP portion and discarding the rest. Later in the reaction, this AMP is transferred to the Okazaki fragments and then is released when the fragments are joined.
DNA ligases can be used to glue together other fragments of DNA, not just Okazaki fragments. It is important as DNA can break from environmental hazards, including ionizing radiation and free radicals. The ligase is used to glue together these fragments to prevent serious dangers from occurring in the body.
DNA ligase is also used to further advance medical research and technology. DNA ligase allows for researchers to break and synthesize DNA strands, creating new genes and genomes.